Since infants cannot provide researchers with verbal feedback necessary for them to know accurately in what their response is to a particular stimulus and what it means to them, researchers have devised these techniques into what they believe are accurate markers of the infant's perception. Each of these methods compares an infant's state prior to the introduction of the stimulus and then with its state during or immediately following the stimulus. These techniques provide the researcher with hard evidence that the infant can detect or discriminate between stimuli. For naturalistic observations, these are the research methods in which infants are observed in their natural environment. There are limitations to this method, being that observations can be unreliable, responses are difficult to quantify due to the rapidness of the movement, and it's not possible to be certain that the infant's response was due to the stimulus or to a change in no stimulus going to a stimulus. So care must be taken not to overgeneralize the data being taken from these observations. So for video analysis, this permits researchers to carefully study the infant's responses over and over in slow motion. Precise measurements can be made of the length and frequency of the infant's attention between two stimuli. So for the heart and respiration monitors, this provides investigators with the number of heartbeats or breaths taken when a stimulus is presented. Numerical increases are used as quantifiable indicators of heightened interest in a new stimulus. So non-nutritive sucking are devised apparatuses that are connected to a baby's pacifier to a counting device. The stimulus is presented and changes in the infant's sucking behavior is recorded. The increased number of sucks were used as an indicator of the infant's attention or preference for a given visual display. For the habituation and dishabituation, a single stimulus is repeatedly presented to the infant until there is a measurable decline. And at this point, this is habituation. A new stimulus is presented, and then this is when the dishabituation in the responsiveness is recorded. So if the infant fails to dishabituate and continues to show habituation with the new stimulus, it will be assumed that the baby is unable to perceive the new stimulus as different. This is used primarily with the studies with infant auditory and olfactory perception. So evoked potentials are those electrical brain responses that may be related to a particular stimulus because of where they originate. Electrodes are attached to the infant's scalp to measure the changes in the electrical pattern of the brain. This indicates that the stimulus is getting through to the infant's central nervous system, and this is eliciting some form of response from the infant.